What if the real face was neither of these? And what if science could actually show us something closer to the truth? Parabon Nanolabs' Snapshot DNA Phenotyping system asks whether a genetic sample can yield a statistically reliable portrait. The process begins in the lab, where DNA is extracted and prepared under strict protocols to avoid contamination. For both forensic and archaeological samples, a minimum call rate of about 70% across a panel of over 1 million single nucleotide polymorphisms is required. This threshold ensures that predictions remain within responsible limits, and below it, results become too uncertain to report. After passing quality control, samples undergo high-throughput sequencing. The resulting genetic data are compared to large reference panels, with missing genotypes filled in by advanced algorithms. Each trait, such as eye color, skin tone, and hair color, is predicted independently based on established genotype-phenotype links. Pigmentation traits show over 80% accuracy, as demonstrated in blind tests at the University of North Texas Health Science Center, where predicted skin, eye, and hair color matched photographs in most cases. Structural traits like nose width and jaw shape are more difficult. Their genetics are complex, and reference data are less complete, especially for non-European ancestries. Confidence scores for these features often fall below 60%. To address this, Snapshot provides exclusion ranges of 90% or higher for traits that can be reliably ruled out, and explicit probability intervals for those that cannot. Snapshot's credibility comes from its record in forensic cases. In the April Tinsley case, the system generated a composite sketch from crime scene DNA that produced new leads before a suspect was identified. Each prediction is grounded in statistical evidence, with confidence intervals disclosed for every trait. This transparency sets a clear standard for what DNA can and cannot reveal, a critical step before applying the method to ancient genomes. 73 ancient genomes, recovered from Bronze Age Canaanite burials across the southern Levant, now form the most complete genetic baseline for the world described in Genesis. These individuals, drawn from sites like Tel Megiddo and Beit Shean, were sequenced using protocols designed to overcome the severe DNA degradation typical of the region's hot, humid soils. Lab teams prioritized the petrous portion of the temporal bone, extracting fragments that often preserved enough endogenous DNA for high-throughput sequencing. Each sample underwent partial UDG treatment to limit post-mortem damage while maintaining authentication markers. And hybridization capture was used to enrich for over 1 million SNPs, including those relevant to pigmentation and facial prediction. Population modeling of these genomes revealed a two-source admixture, with about 60% of the ancestry tracing to local Neolithic Levantine farmers, and another 30% linked to migrants from the Zagros Mountains. Minor contributions from Egyptian and steppe populations were detected, but they had little effect on the alleles that influence visible traits. The frequencies of key pigmentation, SNPs, in this dataset, such as SLC24A5 for skin tone and OCA2-HER-T2 for eye color, allow for statistical predictions at the population level. The majority of these ancient individuals carried the derived SLC24A5 allele, associated with olive to medium skin and the ancestral OCA2-HER-C2 combination, indicating a high probability of dark brown eyes. Red hair alleles were rare, and dark hair predominated. Dr. Ellen Graytack, Director of Bioinformatics at Parabon Nanolabs, leads the adaptation of forensic phenotyping pipelines for ancient DNA. Her team's approach begins with rigorous quality control. Only samples with sufficient SNP coverage enter the prediction models. For these Bronze Age Canaanite genomes, the available data support high confidence estimates for skin, hair, and eye color, while predictions for facial structure remain less certain. This population-level genetic portrait stands apart from centuries of artistic tradition, grounding any attempt at visualizing the patriarchs in measurable evidence rather than inherited convention. Heat, humidity, and time break ancient DNA into short, damaged fragments. In the southern Levant, where Bronze Age patriarchs once lived, these conditions push the limits of modern sequencing. Even with advanced protocols, partial UDG treatment, hybridization capture, and rigorous contamination controls, most samples yield only a fraction of the original genetic material. Dr. Ellen Greytak, a computational bioinformatician, explains that much of the DNA is simply lost to the environment. What survives is patchy, with gaps in the very regions used to predict facial structure. The science behind DNA phenotyping is built on statistical models trained mostly on European and East Asian reference panels. This creates a bias. When the system encounters genomes from ancient Canaan, its predictions for traits like nose width or jaw shape become less reliable. For these features, confidence scores often fall below 60%. The algorithms do better with skin, eye, and hair color. But even here, the absence of Middle Eastern-specific alleles can skew results. In some cases, the model may overestimate lighter features, or underrepresent the range of variation found in the ancient population. Snapshot-style reconstructions are therefore best understood as probability clouds, not portraits. Each trait, nose breadth, cheekbone projection, chin shape, comes with a range of possible outcomes. For Bronze Age Canaanite samples, the most the data can offer is a population-level approximation, a likely set of features, each with its own margin of error. These limits are not unique to biblical reconstructions. Similar challenges appear in attempts to model the faces of ancient Egyptian mummies, or even Denisovans, where scientists now present probability distributions instead of single images. The strongest claim science can make is not a definitive likeness, but a careful, statistically grounded estimate, one that respects both the power and the boundaries of the evidence. Recent years have seen the first practical demonstrations of DNA-based facial prediction on ancient remains. In 2021, Parabon Nanolabs released reconstructions of 3 Egyptian mummies from Abusir el-Malek, using low-coverage genomes recovered by the Max Planck Institute and the University of Tübingen. Their workflow began with hybridization capture to enrich for over 1 million forensic SNPs, followed by imputation and trait prediction, using the same statistical models applied in modern forensic cases. The resulting faces were not single, definitive images, but probability-driven composites, each trait rendered as a statistical best guess, with confidence intervals disclosed for skin, eye, and hair color. For features like nose width and jaw shape, the output was a range of possibilities, visually represented as a probability cloud rather than a fixed portrait. A similar approach shaped the recent Denisovan facial model, where researchers used DNA methylation patterns to infer shifts in craniofacial development genes. Instead of a single reconstruction, the team generated thousands of simulated faces, producing a consensus image surrounded by a halo of variation, the visible expression of uncertainty. Forensic artists and museum curators now work alongside geneticists, integrating these probability clouds with traditional skull-based reconstructions where possible. These cases demonstrate that even with ancient and degraded DNA, it is feasible to generate scientifically grounded, population-level approximations of appearance. Yet each attempt makes clear that the output is a spectrum of likelihoods, not a historical likeness, an advance in method, but one that respects the boundaries of the evidence. Centuries of art have shaped how the patriarchs are imagined. Renaissance painters like Michelangelo and Titian gave Abraham and his descendants fair skin, light eyes, and features drawn from their own surroundings. In Ethiopian churches, icons show the patriarchs with deep brown skin and Afrocentric faces, reflecting the beauty and dignity of local communities. Islamic tradition often avoids faces altogether, using calligraphy or abstract symbols to honor the sacred by refusing to depict it. Each approach reveals more about the artists and their times than about the men themselves. Now, DNA-based reconstructions offer a new kind of image, one built from population averages and probability, not imagination or tradition. For some, this can be unsettling. In a 2019 Pew survey, 39% of American Christians say the Bible should be taken literally. For them, a face reconstructed from Bronze Age Canaanite genomes might feel like a promise of certainty or a threat to cherished beliefs. A faith elder reflects on this tension. A scientific model can never capture the spiritual reality of our ancestors, but it can remind us that they were real people, living and struggling in a real world. The emotional weight of a probability-driven portrait is heavy. It asks believers and skeptics alike to hold mystery and evidence together, to see the patriarchs not as icons or abstractions, but as human beings shaped by history, genetics, and faith. Technical progress continues to narrow the gaps in DNA-based facial prediction. In the ancient DNA lab, researchers now focus on the petrous portion of the temporal bone, a dense structure that often preserves 10 to 40 times more endogenous DNA than other skeletal elements. This single advance has raised the number of Bronze Age samples with usable genetic data, making population-level reconstructions more robust. Hybridization capture methods enrich for over 1 million single nucleotide polymorphisms, including those most relevant to facial and pigmentation traits, even when total DNA is scarce. Dr. Ellen Graytack, who leads the phenotyping team, points to the steady expansion of reference databases as another key development. By adding more Middle Eastern and North African genomes, the models can correct for biases that once skewed predictions toward European features. Polygenic risk models, which integrate thousands of small genetic effects, are also improving the accuracy of structural trait estimates. These combined steps promise to reduce uncertainty, though the results will always be probabilistic, a range of likely faces, not a single, certain image. The science moves forward, quietly refining its methods, one bone and one dataset at a time. Today, Parabon's algorithms and Bronze Age Canaanite genomes yield only statistical averages, never portraits, never certainty. As DNA phenotyping advances, the familiar faces of tradition stand on shifting ground. Science redraws boundaries, but reverence and imagination still fill the space between probability and belief. Share your thoughts below.